Great. So yesterday's lecture was a whistle-stop tour through all the different ways we define biodiversity and all the different ways we think about biodiversity. And I kept hinting at why we're so interested in biodiversity and why we want to quantify and define and monitor biodiversity. And we're going to continue to talk about this in this lecture where we start to think about the functional consequences of biodiversity. I spent the whole of the last lecture explaining to you all the different ways we count biodiversity and all the different indices that we have developed to assess biodiversity. But I didn't really prove to you that biodiversity is important for ecosystem functioning or ecosystem health. Remember the example of the fynbos with its nearly 8,000 species. Would the fynbos function differently or be less healthy if it had only 4,000 species? These are the sorts of questions we need to consider. Do we really need all these species to have functioning ecosystems? Of course, nature lovers are devastated if just one species goes extinct. But policymakers and general society sometimes ask for more evidence to justify efforts to conserve and protect species richness. So ecologists think that biodiversity is related to very important ecosystem properties, such as productivity and stability, but they have struggled to prove it over the years. I have talked a lot in this lecture and previous lectures about functioning ecosystems. So now I'm going to define a branch of ecology that focuses on understanding and quantifying ecosystem functioning. In particular, I will introduce you to some theory about how biodiversity affects ecosystem functioning. If you do my third year course next year, then you will explore these ideas further. So the Wikipedia definition of functional ecology is that it's a branch of ecology that focuses on the roles or the functions that species play in the community or ecosystem in which they occur. So it emphasizes characteristics of the organisms, physiological, anatomical, and life history characteristics that define their function. Some key ecosystem functions that people have identified are things like primary productivity, pollination, because unless plants can pollinate each other, then they can't reproduce. Nutrient cycling, we've talked a lot about nutrient cycling, they're important functions. Water infiltration. Really, the ecosystem function of interest depends on your question. Two key ecosystem functions that people have tried to link to biodiversity are productivity and stability. In the last 2 or 3 decades, people have done many experiments, and they've generally found that you get when you get higher biodiversity, you actually do get higher productivity of plants. It only really happens if you move from like 1 to 2 to 3 species. Once you've got more than about 10 species in an ecosystem, you don't find that more species increases productivity. But definitely there's a positive relationship at very small numbers between the amount of species and the productivity of the ecosystem. The argument is that many different functional types can make more complete use of the resources on offer because they use different niches. Interestingly, if you plant millies at a site, you can probably usually get higher productivity than if you had a natural grassland. But then from year to year, if you planted millies, you wouldn't get the same productivity that you would if you had a natural grassland because it changes from year to year. So overall, you still find that polycultures or mixed groups of different types of species have higher productivity. So this is a general graph. The graph on the left shows the number of species present. And as you increase the number of species present on the x-axis, then on the y-axis, the net primary productivity, which you remember we defined in the carbon cycle lecture, increases. And why is this? It's mostly due to niche separation. So we call the graph on the left the biodiversity productivity relationship. The other thing that people have found when they've done experiments and studied this is that diverse communities vary less in their abundances from year to year than communities with very few species. Communities with very few species show boom and bust dynamics. That's what I was mentioning before. Some years they can do wonderfully, and then other years they can do hardly any production at all. This is sometimes called the insurance hypothesis. When people invest in the stock market, They like to invest in lots of different stocks because some of the stocks do well in some years and some of the stocks do well in other years. So overall, if you invest in lots of different stocks, you have a stable investment structure. And it's the same with organisms. If you have many different organisms in a system, then overall from year to year, you have more stable ecosystems. So people call this the biodiversity resilience relationship, or the biodiversity stability relationship. Again, on the x-axis, you have the number of species present, or functional types present. And on the y-axis, you have the variability of net primary productivity. So how much it varies from year to year. If you have just a few species, then it's very variable. As the species increase, then the variability decreases, because in some years one species does well, in some other years another species does well. So you need to be able to look at these two graphs. You need to be able to describe the functional relationship. So as the value on the x-axis increases, what happens to the value on the y-axis? That's the functional relationship. So you need to describe the functional relationship between species richness and net primary productivity. And you need to describe the functional relationship between species richness and the variability of net primary productivity. Then you also need, once you've described the relationship, you need to explain the relationship. So these two terms should help you to explain the relationships that I showed you in the slide before. So after all these experiments were done, and people could start to explain how biodiversity linked to ecosystem function, they coined these two terms. So there's the concept of functional complementarity. Functional complementarity is when species are functionally different so that they do different things. So if there's functional complementarity, then communities are more likely to make efficient use of available resources, because one species uses this resource and the other species use the other resource, and overall you get more productivity. Then there's the idea of functional redundancy. That's when different species function in the same way. They can be substituted for each other. So communities that contain functional redundancy are resilient to losses in an individual or a few species. They can still function. So communities that have functional redundancy are more stable. So these two terms, complementarity and redundancy, help you explain the patterns that we saw on the slide before. A real-world example of functional complementarity would be plants that have different rooting depths. So if there was only one plant rooting at one depth in the soil, then the water resources from all the other depths would go unused. However, if you have 3 or 4 different plants, each with different rooting depths, then the plants can make use of more of the water in the soil, so you end up with higher productivity. You get the same patterns with herbivores browsing food plants. You might have heard in first year or even in Jason's lectures around the different niches that different herbivores use. In particular, in our savannas, we've got giraffe that browse at about 3 or 4 meters of height, and we have small little impala that browse at below 0.5 meter. And so because the herbivores browse at different heights, they can make use of more of the forage resources. So you end up with higher productivity of herbivores. I mentioned that people had done lots of different experiments to prove these relationships. And this is an example of just one experiment that was done. Dave Tillman is a biologist from Minnesota who set up a huge experiment where he planted different numbers of species of grasses and forbs in hundreds of small plots on the prairie of the USA. You can see how big this is. It's kilometers across this experiment, and each little patchwork in this landscape consists of a plot with different numbers of species in it. His original results demonstrated that plots with more species had higher productivity and were also less variable from year to year, as we mentioned above. So his data confirmed the idea that biodiversity increases productivity and resilience. However, more recently, people have reanalyzed his data, and they've showed that it was actually plots with higher number of different types of species that showed higher productivity and resilience. So you get better relationships when you consider functional richness as well as species number. And we talked a bit about the difference between functional diversity and species diversity in the previous lecture. So this experiment is a good example that shows that what matters is not so much how many species you have, but how many kinds of species you have. And this picture at the bottom of all these people poring over these plots is just to show you how Dave Tillman managed to collect all that data. He got all his undergrad students to work on his plots all the way through the summer, and that's how he managed to run this big experiment. As I've mentioned, this has been quite an active area of research and discussion for the last couple of decades. So just to summarize, most researchers these days argue that functional diversity is more relevant than taxonomic diversity. And there are some references that you can read if you're interested in understanding this debate. All right, so that's fine. I also agree that it probably makes more sense to look at the functional diversity than the species richness. But how do we go about quantifying functional diversity? It's very easy with the help of a plant or an animal identification book to quantify species richness. You just count the number of species. What methods can we use to quantify how different these species are from each other? To do this, we have to measure things about the species. We have to measure their functional traits. And the functional traits are characteristics, morphological, biochemical, physiological, behavioral characteristics of organisms that influence their performance or their fitness or how they work. And these traits are used to define the ecological roles that species have in an environment. Now, because the traits reflect the function, there are sometimes trade-offs in terms of the types of traits that an individual organism can display. For example, you can't be a big tree and capture lots of light if you also want to be able to be resilient to fire. So Life History Strategy Scheme describes the syndrome of traits that result in an organism. So we measure functional traits, but then we end up defining something called functional types. So the different types of organisms that you find in a system. And people believe that the kind and the abundance and the range of traits present in a community affects ecosystem processes. As I mentioned, we measure all these functional traits so that we can group species into functional types. You can think of a functional type as like a functional characteristics of a species. It's like, let's take these 20 species. For example, in the fynbos, we had 8,000 species, and they got grouped into 4 different functional types. So functional types are groups of species that are ecologically similar. And functional types depend on the question being asked. You can't just, anyone who's coming into a system will see different functional types there, and that's what makes it a bit more complicated. Another thing people consider a lot when they're trying to look at the functional consequences of biodiversity is dominance. So I have a field site in the Cradle of Humankind near Johannesburg. There's a picture of it with some of my students, and it's a grassland, and it's beautiful, and it's packed with interesting grass and forb species. And on the left-hand side of this slide, you can see a list of all the different forb species, the wildflower species that I found at my site over the years. And you can see that although we have 44 recorded species, most of them have only been recorded once or twice. Only 2 of them are abundant. Acalipha angustata, which is this little pinky purple thing on the right-hand side, and Senecio venosus, which is the daisy on the left-hand side. So we say that these 2 species are dominant because they account for most of the biomass in the wildflowers on my plot. We can use what we call a rank abundance curve to visualize patterns of species richness and evenness. And when we plot the rank abundance curves for ecosystems across the globe, we find that most communities are actually dominated by a few species. So have a look at the graph, which shows the rank on the x-axis. So that's the number of species in the order of the abundance. So the most abundant species are ranked first. And then on the y-axis, it's the relative abundance. And we come back to the same question. Is the orange or the brown community more species rich? And is the orange or the brown community more even? You need to make sure that you can interpret this graph and you can understand the concept of dominance. So there would be people who would argue that my site in the Cradle of Humankind, my field site, isn't very diverse at all because it's dominated by only 2 different species. But other people argue that just because a species is rare doesn't mean it isn't essential for ecosystem functioning. So this Few species, so some rare species, low abundant species, can still play critical roles. So pollinators, like the butterfly on the left, or biocontrol agents, like the ladybird on the right, are both vitally important for healthy functioning ecosystems. Without the right pollinators, then the plants which dominate the biomass would not be able to reproduce. And without biocontrol agents, the ecosystem could end up devastated by pathogens and pests. So people argue that you can't just consider the dominant species when you are trying to understand ecosystem function. And for this reason, because there are examples where rare species are very important, we're going to learn to contrast the difference between a dominant species and a keystone species. A dominant species is the most abundant species present in an ecosystem, makes up the highest percentage of total biomass. So for example, the way you quantify that is you do your rank abundance curve and you say, how quickly does that rank abundance curve drop off? And you quantify the dominant species. So then, of course, dominant species have a big impact on ecosystem functioning because they're using most of the resources and they make up most of the biomass. But then people have also coined the term keystone species. Now, a keystone species is a species that has a disproportionately large effect on its environment relative to its abundance. So if it is removed, it would have big impacts on the way that the ecosystem functions. We identify keystone species by observing the changes that happen to ecosystems when these species are removed. We call these consequences cascading effects. You could also call them domino effects. It basically means that if you remove a keystone species, it can affect many other parts of the ecosystem through knock-on impacts on the abundance or dominance of other species. Let's think of some examples. Elephants have big impacts on ecosystems. We all know that. When you have more elephants in a system, they can totally change it. They can take a forest and knock it down and turn it into a grassland. But elephants are also a dominant species. They make up most of the herbivore biomass. When I was doing these analyses, looking at herbivore communities across Africa, we always had to plot our maps with and without elephants, because if you add elephants, then you can't see any of the other herbivore biomass because elephants are so dominant. So elephants are dominant species, but pollinators can be keystone species without being dominant. They can make up hardly any of the biomass, but if you lose them, then the entire ecosystem can collapse. Similarly, many predators can be keystone species. So as we mentioned, predators are quite far up the food pyramid, so there aren't a lot of them compared to the number of herbivores and the amount of plants. But if you remove a predator from a system, then you can get big impact because then nothing's controlling the herbivore numbers, so the herbivore numbers increase. And if the herbivore numbers increase, then you end up decimating the plant populations. So you can have cascading impacts on ecosystems because predators are keystone species. Finally, termites are often considered keystone species because although they are very small and don't make up a lot of biomass, they can engineer the ecosystem, they can change the soil conditions, and they can change the landscapes. This is an example where adding just one extra species to a system totally changed its functioning. The species was a grass, it was called Bromus tectorum, that invaded a shrubland where there was hardly any grass before. If you look at the picture and you take away all the yellowy grass color, you'll imagine what this landscape was like before. It was a very arid shrubland with a lot of bare ground. Then the grass invaded and it filled in all the gaps in the system. The system now burns in fires every few years, whereas before it hardly ever burned. Species richness at this site has hardly changed at all, but the functional diversity has increased and the functioning of the system has totally changed. I showed you this slide in the energy balance lectures as an example of how different landscapes in the same environment can have very different ecosystem properties and energy balances. So this fence line contrast's difference between the two landscapes is due to the loss of just one species called Portulacaria africana. If you look on the left-hand side and count the dark blobs of dark green trees, those are acacia trees, there's actually the same number of trees in each landscape. What's missing is the Portulacaria africana, that pale greeny yellow shrub thicket that's in between them. This is a picture of Portulacaria afra, the speckworm, and the loss of just this one species can convert a dense succulent thicket into a savanna. So it's another example where the addition or taking away of just one species can alter the ecosystem functioning. This lecture has been getting you into some of the real nitty-gritty of ecology and how people kind of grapple with and try to engage with big questions around how biodiversity drives ecosystem functioning. There are a couple of things I need you to be able to do by the end of this lecture. You need to be able to define what is meant by a rank abundance curve and to discuss the importance of dominant species to ecosystem functioning. You also need to be able to describe the ways that people have demonstrated links between biodiversity and ecosystem function. So these are the graphs that I showed you, the biodiversity productivity relationship and the biodiversity stability relationship. You need to be able to distinguish between a keystone species and a dominant species. And you need to be able to discuss methods that people have used to quantify functional diversity and the ways that people group species into functional types. Finally, you need to be able to engage in this debate that's been going on in the literature about the importance of species richness versus functional richness in ecosystem functioning.